Hello, everyone. Today I'm going to talk about my research into phenotypes and brain function in complex social insects. One of the big questions I'm interested in is the effect of social life on brain evolution and brain plasticity. Here you can see some examples of the diversity found in social primate brains of different species. Interestingly, a similar level of size diversity can be found within brains from a single species, one which is also highly social. These are brains from leafcutter ants, and they'll be the focus of my talk today. The specific model that I'll be using to examine social brain evolution is the leafcutter ant Atta cephalotes. Leafcutter workers can vary greatly in size, as you can see here, and there's a pronounced division of labor in this species with different workers specializing in different tasks. They engage in large-scale agriculture where they farm their own symbiotic strain of fungus, which looks like this, by supplying it with leaf mulch. The advent of agriculture is thought to have had significant impacts on human brain evolution, and it is therefore useful to study social brain evolution in a model that also relies on farming. We first wanted to observe workers in this species to assess their behavioral differences. We made observations by dividing up a lab colony footprint into a grid, and for 10 minutes at a time, we looked at one square and recorded the size of each worker that was inside it and every behavior that they performed, and we did this twice for each square in the colony. I just wanted to show you the results for two of the key behavioral categories most relevant to agriculture in this species. We saw significant differences in leaf harvesting task performance, which includes selecting, cutting, and carrying leaves among the worker groups, with the media workers performing these behaviors most commonly, and the frequency distribution for this task category is quite narrow. And here's the same plot, but now for fungal gardening, which includes mulching, fertilizing, and pruning the fungus. This distribution skews much more towards performance by minims and smaller media workers, and this set of tasks also differed significantly among worker groups, with the second smallest group performing it more than all others. We also wanted to know how flexible different types of workers were in terms of performing a variety of tasks, and to assess this, we used the Shannon Diversity Index. This showed that the second and third smallest workers, which are both in the media size range, performed a broader array of tasks than did the smallest minim workers or largest major workers. Next, we wanted to look at differences in brain anatomy among workers. I want to first describe two models that have been used to understand patterns in which brains evolve. When overall brain size is a strong predictor of neuroanatomy within a given group, this is consistent with concerted evolution, meaning brain evolution constrained by developmental processes that link neuroanatomy to body size or that link together developmental trajectories of different brain compartments. And when there is instead a lot of diversity in the proportion of different brains occupied by different functionally specialized compartments, and it's not simple enough to be predicted from brain size, this type of scaling is called mosaic brain evolution, or independent selection on different areas of the brain. And these two models are not completely opposed to each other, but rather are different approaches to weighing the impacts of various forces on brain scaling, and they can co-occur in the same system. Another way of forming hypotheses about brain evolution is to consider the behavioral challenges faced by different organisms based on their sensory environments and cognitive processing requirements. So to do that, we went for a novel approach that takes more specifics into account than the previous models. I came up with this simple formula, which is just multiplying a score of sensory integration demands by a score of task performance frequency, and then adding up those products. And these scores were generated in part from our previous observations and in part from existing literature, and we attempted to consider all behaviors performed by workers in this species. The resulting number represents the expected level of investment in brain tissue based on the degree to which sensory integration is required to accomplish a given task known to be performed by different workers. The sensory integration demands that our model describes most closely correspond to the function of a brain region called the mushroom bodies, which mediate learning and memory and process inputs from different sensory areas of the brain. And now let's look at the hypothesis generated by our model for estimating sensory integration requirements in the brain, specifically in the mushroom bodies. The y-axis here is the estimated score for neural requirements based on our equation, and the x-axis is worker size. As you can tell, media workers are estimated to require more sensory integration processing than minims and much more than majors, and this is largely a reflection of their behavioral diversity. And here's the observed pattern of mushroom body investment. This area was significantly larger in media workers in terms of the proportion of the brain it occupied, and this allometric scaling might help media workers remember where to find leaves when they're out foraging, among other things. And visually, there's a very strong resemblance between the predicted and observed patterns. And when you perform a linear regression, the relationship between the two is significant, but only a moderate amount of observed variance is explained by our predicted estimate. This supports the idea that selection on brain compartment volume optimizes for task integration requirements, though other factors likely influence levels of investment. And we next had further questions about the molecular mechanisms behind observed mosaic brain phenotypes. So in looking at functional mechanisms, we elected to focus on brain gene expression, since workers in this species are sisters and raised in the same environment, so we wanted to look at a dynamic regulatory process. If expression patterns are mostly linked to overall worker size and brain size, then almost all genes should predictably increase in expression in larger workers, or they should mostly decrease linearly in larger workers. Whereas influence from mosaic evolution would make us see all kinds of different patterns, and not all expression should linearly correlate with increasing worker size. So to test this, we measured whole brain gene expression in minims, medias, and majors using RNA-seq, and we found significant differential gene expression, with most of the differences being between majors and minims. And when we performed principal component analysis on our data, as you can see here, we saw that principal component 1 explained 45% of the observed variance, and it looks like each of the size groups are spread out in order along that axis. As you can see in this visualization, PC1 has a strong correlation to worker size, while PC2 is more constant. So PC2 is likely being driven by some other trait or force. And we saw in the previous figure that PC2 explains 20% of the variance. So this means that there's a substantial chunk of variance that is being driven by some trait that neither positively nor negatively correlates to increasing worker size. And this provides some evidence for influence from mosaic brain evolution as well. To explore the functions of genes in our overall data set, I performed gene set enrichment analysis to look for overrepresented gene ontology terms which reflect gene function. And I saw that numerous metabolic process Go terms were overrepresented, but were mostly enriched in genes upregulated in majors. And several sensory-related terms were more highly enriched in genes upregulated in either minims or medias. And so to conclude, I'd like to thank my advisors, my lab members, and funding sources. Thank you for your time.